Welcome to all viewers and well-wishers of the world. Purpose of my lecture is learn the knowledge from the word, return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot. Now it's time to return a lot to the word from where I have got all this knowledge. The topic of today is exotoxin and endotoxin. First, what is toxin? Toxin is a poisonous substance produced within the living organism, inanimate, not capable of reproducing themselves, not produced artificially, was first used by Ludwig Brigger, derived from the word toxic. Toxins are the small molecules, peptides, or proteins. They are capable of causing disease on contact with or absorption by the body tissues. Toxins vary in their toxicity, minor like bee sting to the deadly botulinum toxin. Toxins are different from other chemical agents by their method of production. It simply means it's a biologically produced poison. So the classification of the toxins, there are two main types, exotoxin and endotoxin. Both gram-positive and gram-negative bacteria secrete exotoxin, which are the proteins, but endotoxins are only produced by lipopolysaccharides, LPS, not secreted by the gram-positive bacteria. So the LPS of the gram-negative bacteria is the endotoxin. These are the integral component of the cell walls of the gram-negative bacteria only. So the exotoxin first, exo means outside, produced by the viable microbes, but excreted outside of the microbes. Microbe just make it and excrete outside. The function of the exotoxins for bacteria are usually unknown. Why they are being produced by the bacteria in their cytoplasm is unknown. They are capable of destroying or injuring host cells only. That include many types of proteins and the enzymes in nature produced inside the bacteria as part of the growth and metabolism, then secreted or released from the microbes before mostly or after lysis. They do excrete and secrete these toxins when they are alive or they are just dead or lies. They do release the exotoxin. These exotoxin diffuse readily into the surrounding medium or the tissues. Here one can see the bacteria like Clostridium botulinum. These are secreting the exotoxins. Now the host cell have got two kind of receptor. One is the exotoxin receptor. The other are the antibodies. If the exo, if the exotoxin do combine with their receptor, and here one can see the exotoxin receptor exotoxin interaction, and that causes the death of the cell. Other, Otherwise, if the exotoxins are being taken up by the antibodies on the surface of the somatic cells, these antigen and antigen antibody interaction occurs and the host cell is able to live and survive. So here one can see the immune response to the exotoxin. These are the exotoxin and these are deactivated exotoxin by the application of certain chemical. So these activated exotoxin become just deactivated exotoxin, which are not capable of causing any problem, but these are converted to the toxide. They are not capable of causing disease, but they are immunogenic. They are capable of inducing or just initiating the antibodies and not going to die. As the cell is going to die because of the receptor and the exotoxin interaction, but they are going to become toxide. So exotoxin can diffuse within blood and travel throughout the body. They destroy parts of the host cells or inhibit the metabolic function of the host cells. In host, if these uh, uh, exotoxin in the host survives, the host produce neutralizing antibodies or antitoxin to neutralize the effect of the exotoxin to survive the somatic cell. As exotoxins are excellent antigen capable of generate specific antibodies called antitoxin, exotoxin can be destroyed or inactivated by dilute formaldehyde, iodine, and the other chemicals. Here one can see the bacteria and the cytoplasm of the bacteria is just secreting the exotoxin, and one can see these exotoxin secreted by these bacteria are going to affect the different tissue and the target organs like heart, lung, and other body tissue. So these are going to just destroy these tissue by the exotoxin. Similarly, if the exotoxin receptor is there and if that exotoxin receptor is going to be taken up by these host cell, that is going to destroy the host cell, and if these are being taken up by the antibodies, so these are not capable of destroying the host cell. Here again the target host cell and the antibody molecule, one can see these are going to catch the exotoxin and just neutralizing the exotoxin, and the exotoxin is not capable of just going to attach to the receptor which is for the exotoxin on the host cell. Similarly, receptor here, these are not being occupied by the exotoxin, but the antibodies get it attached and make it neutralize. All these chemicals destroy the toxic activity of most of the oxytocin, as I have told you, the phenol, the formaldehyde, but they do not, but they does not affect their antigenicity. Means they are still capable of producing antibodies and behaving like antigen. Inactivated toxins are called immunogenic toxides, are useful in preparing vaccine like the tetanus toxide. Exotoxins are the soluble polypeptide proteins. Mostly higher molecular size exotoxins are heat labile. They are inactivated at 60 to 80 centigrade, and so they are not capable of causing damage to the host cell. Low molecular size exotoxins are heat stable like staphylococcus and enterotoxin, and they can damage the host cell. So here one can see the toxide formation, the diphtheria bacteria producing the toxin, and these bacteria toxins are inactivated to make just the diphtheria toxide, which is capable of producing the antibodies. So the diphtheria toxin modified to the diphtheria toxide. Similarly, one can see the the toxin, and toxin under the chemical modification, they become toxide. As you can see, the toxin moiety of the toxin that have been detached, and that is just now toxide, and that is antigenic determinants because of loss of toxin moiety. Exotoxins are highly potent in minute amounts. 1 microgram of the tetanus exotoxin can kill adult human. 1 milligram of the tetanus or the botulinum toxin can kill 1 million guinea pigs, and 3 kilogram of the botulinum toxin can kill all the population of the world. Here one can judge the toxicity of these exotoxin. Exotoxin proteins are mostly encoded by the genes carried on the plasmid or the template bacteriophage. Each toxin produces typical effect with characteristic clinical manifestation. Some gram-negative organisms like Shigella, cholera, Vibrio, and E. coli also produce exotoxin, though they are capable of producing endotoxin, but they are also capable to produce exotoxin. Now the groups of the exotoxin are of following 5 types based on their biological effect on the host cells, and their name are also self-explanatory to tell where they do affect: cytotoxins, neurotoxins, enterotoxins, exoenzyme, toxic shock syndrome toxin, TSST1. I am going to tell you detail of all these toxins one by one. First, the cytotoxin. Large group means they are capable of causing damage to the cytoplasm of many host cells. So the name is cytotoxin. It has got specificity for many host cells. Example is diphtheria toxin produced by the Corynebacterium diphtheriae. It inhibits protein synthesis in the many host cells. They cause damage to the variety of the cells, disturb cell function, or cause cell lysis. The number 2 types of the exotoxin are neurotoxin, means cause damage to the nervous system. Major symptom is paralysis. Example is toxin produced by the Clostridium species like botulinum toxin by the Clostridium botulinum, tetanospasmin by Clostridium tetani, and Shiga toxin. These are all neurotoxin. They act on the motor neuron, prevent release of the acetylcholine at the myoneural junctions, so prevent muscle excitation, stimulation, and producing flaccid paralysis. There are certain things which we cannot buy the money which we do have, and that is the time, happiness, friends, dreams, hope, love, and the health. So take care of all these things, as these cannot be bought from the market. Now, number 3 is enterotoxins, which damage to the intestines and the tissue of the digestive tract. Major symptom is vomiting and diarrhea. These stimulate hypersecretion of the water and electrolytes from the intestinal epithelium and produce watery diarrhea. Mechanism by which they do it is unknown. Perhaps inflammatory response to the bacteria or the toxin activate neurological control mechanism to regulate water and electrolyte transport and causing the diarrhea and hypersecretion. Some enterotoxin behave as cytotoxic, like Shiga-like enterotoxin from the E. coli. The enterotoxin, some disturb host cell function, but no structural damage to host cell, but they are called cytotonic, like collar toxin. They are only going to disturb the host cell function, not going to kill the host cell. Enterotoxin disturb normal smooth muscle contraction, causing abdominal cramping, and enterotoxigenic E. coli and Vibrio cholerae produce diarrhea after attaching to the intestinal mucosa, where they elaborate enterotoxin. These cause extensive ulceration of the mucosa and rarely enter the bloodstream. The exoenzyme, these are mostly enzyme in nature released by the bacteria, act and damage different tissues of the body as their name indicate. These include protease, phospholipase, hyaluronidase, collagenase, fibrinolysin, coagulase, streptokinase, staphylokinase, exfoliatin, and scalded skin syndrome toxins. And exoenzymes, one can see they are capable of making this gap in the between the host cell by having hyaluronidase, which is going to destroy the hyaluronic acid. Similarly, they are going to destroy the collagen layer between the epithelial cell, and invasive bacteria reach the epithelial surface from this destruction of the collagen layer. Similarly, one can see the bacteria invade the deeper tissue by just destroying these uh, collagen and hyaluronic acid. And the enzymes used for the penetration, these are also the certain enzyme I have told you also, the collagenase, lecithinase, they are able to just penetrate the host tissue and invade it. Here one can see they can go to the deeper layers and can cause the problem. They can enter even into the blood vessels by the streptokinase and the other enzyme like coagulase. So these can enter into the blood vessel by destroying the layers of the blood vessel and by the action of certain enzyme which they produce and causing the damage to the blood vessel and the tissues. Now, fifth one is toxic shock syndrome, toxin TSST1. It causes severe to the fatal problem. Even with the antibiotic treatment, these are not going to be reversed. The action of the TSSTI cause sudden onset fever, diarrhea, vomiting, hypertension, rash, renal and the muscle damage. Two toxins are enterotoxin, TSST1, superantigen and the pyrogenic toxin. These are responsible for causing toxic shock syndrome. So the type of the exotoxin, these are of three principal types: AB toxin, membrane disrupting toxins and superantigen. First, the AB toxin, as the name indicates, it has two parts. The A chain active part has inhibitory activity against some vital function, get enter into the host cell and cause the damage. The other is B chain, name is binding part. It binds to the receptor on the host cell and promotes the entry of the A chain, which is the active part, to cause the damage to the host cell. Mode of action is inhibition of protein synthesis, inhibition of neurotransmitter release, hypersecretion. Toxin gene is encoded on a plasmid or a lysogenic phase. So here one can see the AB toxin, the A subunit responsible for causing damage, the B subunit for getting the attachment. As you can see, they are just getting entry into the bacteria, and in the endosome, this A and B unit get enter, and from here the A subunit, which is responsible for causing the damage to the host cell, that is released from the endosome. Similarly, one can see the binding by the B component, and the active component is allowed to enter into the host cell through the cell surface and to cause the damage. The other type is membrane disrupting toxin. These include proteases, phospholipases, detergent-like action they have. These digest enzymatically the phospholipids or the protein components of the plasma membrane. So these make the way for the toxin and then the bacteria to get enter inside the host cell. These behave as detergent, causing lysis of the host cells and making hole in the plasma membrane by the two ways, form protein channels in plasma membrane, or they disrupt phospholipid portion of the plasma membrane, and the end result is the entry of these bacteria into the host cell. Here one can see the phospholipase exotoxin that is just going to damage these hydrophobic bonds between these two, and ultimately when this bond is gone, so the unstable host cell membrane, as you can see the parts of the host cell membrane, and ultimately the cell lysis and the death occur because of the action of these phospholipase exotoxin released by the bacteria. Similarly, one can see these pore forming exotoxin that make pore protein, make the hole in that, and this just make the passage for the exotoxin to get enter into the cytoplasm, and the exotoxin form the pores in the membrane by their action of certain enzyme. Here, the cytoplasmic contents are out because that is the bacteria and their toxins, exotoxin, cause swelling of the host cell, lysis, and the death of the host cell because of the high osmolarity, and the contents of the host cell leak out and ultimately causing the death of the host cell. And continuing membrane disrupting toxin examples are phospholipid toxin of Clostridium perfringens. It destroys the blood vessel, stopping influx of the inflammatory cells, create an anaerobic environment important for the growth of this strict anaerobe. So favor the growth of this anaerobe. Delta toxin of Staph aureus is an extremely hydrophobic protein. It inserts into the cell membrane and have a detergent-like action, make the hole, and ultimately disrupt the membrane and causing the death. The third variety is the superantigen. These provoke very strong immune response, so the immune system produces too many immune cells that produce fever, vomiting, diarrhea, sometimes even shock and death. That is not because of the exotoxin themselves, but the immune system that has been just excited and causing producing many immune cells which are responsible for all these fever and vomiting and other symptoms. So the most notable exotoxins are diphtheria toxin, toxin of the Corynebacterium diphtheriae, tetanospasmin, tetanus toxin or the neurotoxin of the Clostridium tetani, botulotoxin or botulinum toxin of the Clostridium botulinum, toxin of the Clostridium perfringens, Streptococcus erythrogenic toxin, toxic shock syndrome toxin 1 TSST1, and exotoxin associated with the diarrhea disease.